Hi everyone, my name is Mara Burrows. Today is March 17th, 2020. This is week 2 and lab number 3. The experiment is called dilutions in viable plate counts. So in the instructions for this lab, they had us make agar plates ahead of time, which I have already done so. I have them plated, and there is the date and the type of agar that's on here. I also have all my materials ready for me. I've disinfected my workstation. I have my water, my graduated cylinders, my agar plates, my rack. I have some alcohol, my pipette. I also have the yeast that I have in warm water that is currently activating and is actually bubbling up. So this should be ready by the time we start to pour. So what I'm going to do first is I'm going to go ahead and ensure that my pipette is sterile and clean by dipping it into the alcohol and just kind of making sure it's been clean and dry. So I'm just going to shake off the excess alcohol, set that down. After that, I'm going to go ahead and get my 6 test tube holders, and I'm going to label them from 10-1 up to 10-6. Let's do that. And 1. Okay, everyone, so I've already labeled my 6 test tubes. They're labeled 10-1, 10-2, 10-3, 10-4, 10-5, 10-6. So now what I'm going to do is I'm going to go ahead and get my pipette, and I am going to add 2.25 milliliters of distilled water to each tube. So just about this much. All right, that is done. We've put 2.25 milliliters of water in every single tube. Now what I'm going to start to do is I'm going to go ahead and swirl the yeast just to make sure that the organism dispenses evenly. I am going to pipette 0.25 milliliters of this into test tube 10-1, which is this one right here, 10-1. So the instructions tell me to go ahead and pipe the solution up and down several times just to ensure that it gets everything, which I'm going to go ahead and do. Okay, so I think it's all there. This is what it looks like. Next, I will set that aside. I'm gonna go ahead and take 0.25 milliliters of the 10-1 sample and pipette it into 10-2, and we'll do so for the other test tubes as well. So 10-1 to 10-2. We'll do 10-2 to 10-3. Ten three to 10-4. Which is actually barely any drops. 10-4 to 10-5. And then 10-5 to 10-6. Okay, so they have all been passed around. What I will go ahead and do is then rinse my pipette off in the distilled water to make sure all the organisms are gone. And now we'll start inoculating the plates. To do that, I already have my agar. I'm gonna go ahead and label the very bottom of the agar accordingly. So it'll be 10-1 to 10-6. 10-1, 10-2, 10-3, 10-4, 10-5, and 10-6. Okay, so these are all labeled. Next, what I will do is go ahead and start the inoculating process. So what I'm gonna do is I'm gonna go ahead and sterilize my pipette again with alcohol, making sure that it's clean, drying it off. Okay. And I'll be piping about 4 drops from the 10-1 bottle. Ooh. Into the 10-1 agar plate. Do about 4 drops here. 1, 2, 3. Oh, 1 more. And 4. Okay. I will go ahead and kind of spread it out evenly to make sure it goes everywhere. And cover it up. So this one, 10-1. I'm gonna go ahead and continue to do that to all the following specimens. So 10-2. 3. Ten, 4. Ten, 5. And then we'll do the last one, which is 10, 6. Okay, so all of that has been plated in the agar. What I'm going to go ahead and do is place my agar in my incubation area or top side up for 12 hours and then flip it over and let it incubate for another 36 hours and go from there. Thank you very much.